Welcome back. In session 1 of the epithelial glands, the introduction and classification was discussed. In session 2 of epithelial glands, microstructural organization of salivary glands will be discussed. There are three pairs of large salivary glands, the parotid, submandibular and sublingual glands, and numerous minor mucosal salivary glands throughout the oral cavity, which secrete about 10% of the total volume of saliva. Okay. Now, some of the minor salivary glands are von Ebner's glands in tongue, glossopalatine in palatoglossal and palatopharyngeal arches, buccal glands in cheeks, and the labial glands in lips. Salivary glands secrete saliva, which is composed of water, electrolytes, which includes calcium, potassium, sodium, chloride, bicarbonate, okay, and also the saliva is composed of mucus, enzymes, phosphate and antibodies. The functions of saliva includes digestive function, which includes moistening the food to help in degglutination, dissolving and suspending food, and initiating digestion of carbohydrates by the enzyme salivary amylase present in it. It also has lubricating and protective functions. Protective function is bactericidal, that is controlling bacterial flora due to the presence of lysozyme and immunoglobulin A. With a normal pH of 6.5 to 6.9, saliva also has an important buffering action. Okay, it acts as a buffer. Around 1 liter of saliva is produced every day. The cells of the duct system draining the secretory units modify the saliva with much sodium and chlorine reabsorbed while certain growth factors and digestive enzymes are released and added to the saliva. We will talk about all three major salivary glands in detail from this slide. These glands are classified under exocrine glands. All salivary glands basically have the same structural organization consisting of three components: parenchyma, stroma, and duct system. Parenchyma is formed by the secretory acini and the myoepithelial cells of the gland. Stroma is the connective tissue in which the parenchyma lies. Stroma provides structural framework to the organ. The glandular tissue is completely enclosed or covered by connective tissue sheath called capsule. Numerous septa arise from capsule, pass into the gland, dividing it into lobes and lobules. Blood vessels and nerves pass along the connective tissue septa to reach the secretory elements. Duct system consists of the ducts which convey the secretory product from the gland into the oral cavity. The ducts draining the lobule is called intralobular ducts, okay, lying within the lobule, which includes intercalated and striated ducts. The intercalated duct is the initial duct which is connected to the secretory unit, okay, and these ducts they join to form the striated ducts, and the striated ducts they join to form the interlobular ducts present between the adjacent lobules. The interlobular ducts join to form the interlobar ducts present between adjacent lobes. Interlobar ducts join to form single main duct which opens into the oral cavity. In this slide, let us discuss structure of parenchyma in detail. It consists of two secretory units on a branching duct system arranged in lobules. They are separated by the septa of connective tissue. As discussed in previous slide, parenchyma has two components: secretory units and myoepithelial cells. The first component of parenchyma, which is secretory unit, has two epithelial cell types: serous cells and mucus cells. Serous cells and mucous cells are arranged in the form of acini, okay, which was discussed previously also. So let us recall what are acini and the different types of acini. Acinus is a small sac-like dilation located at the end of the first part of the excretory duct system known as the intercalated duct. If the acinus is lined by serous cells, it is called the serous acinus, and if the acinus is lined by mucous cells, it is called mucous acinus. Remember, we discussed about acini in first part of epithelial glands. Let us recall and see the features of the serous acinus. It is made of serous cells which are pyramidal or triangular in shape. Nuclei are spherical or round in shape, present at lower half of the cell. The apical part is filled with eosinophilic secretory granules called zymogen granules. The cytoplasm at the base of the cell is basophilic. Lumen is small. Serous acinar cells secrete enzymes and other proteins. Okay. So the acinar cells, okay, in the serous acini, they secrete enzymes and other proteins. To recapitulate the mucous acinus, it is made of mucous cells which are tall, almost columnar in shape. The cytoplasm is filled with pale or light stained secretion, the mucus, which is in the form of mucinogen granules with hydrophilic mucin that provide lubricating properties in saliva. Because of these mucus granules, the nucleus is displaced, compressed and flattened towards the basement membrane. The mucinogen granules are lost during the slide preparation and stains pale and gives the frothy appearance. The lumen is large compared to the serous acinus. In some glands, both mucus and serous cells are present in the same acinus, where mucus cells predominate and the serous cells form a crescent or moon-shaped cap over the mucus cells. These crescent-shaped cells are known as the serous demilune. The secretions from serous cells in the demilune enter the lumen through the tiny intercellular canaliculi between the mucous cells. Recent studies shows that the serous demilune is nothing but artifact where the serous acinus is compressed against the mucous acinus due to the swelling during preparation of the slide. The second component of the parenchyma is the myoepithelial cells. The cells which surround both serous and mucous acini between the acinar cells and the basement membrane are the myoepithelial cells. Myoepithelial cells are small, flattened, spindle-shaped cells which extend during contractile processes around the secretory unit or duct like a basket. Because of this nature, they are also called as basket cells. Secretory products are expelled from the acini by the contraction of myoepithelial cells. These cells are present around the alveoli, intercalated ducts, and large ducts of salivary glands, and they help in the secretion. Serous and mucus and the mixed glands. Okay, this is to talk about the excretory duct system. Okay, so these three glands, that is the serous, mucus and the mixed glands, empty the secretions initially into the intercalated ducts, which are the smallest ducts lined by the simple squamous or low cuboidal epithelium and are surrounded by the myoepithelial cells. Several intercalated ducts merge to form larger striated ducts lined by columnar epithelium exhibiting tiny basal striations. The striations are the basal infoldings of the cell membrane and contain the mitochondria and cellular interdigitations. Striated ducts reabsorb sodium ions from the initial secretion, and their folded cell membranes present a large surface area with ion transporters, facilitating rapid ion transcytosis and making the secretion slightly hypotonic. These striated ducts join to form larger intralobular ducts lined by columnar epithelium. The intralobular ducts from each lobule converge and drain into interlobular excretory ducts. Okay, and these join to form the interlobar excretory ducts. Okay, with increasing size and thicker connective tissue layers. These ducts at the terminal part are lined by stratified locuboidal or columnar epithelium and join to form single large terminal excretory duct, which before emptying into oral cavity is lined by stratified squamous non-keratinized epithelium. To discuss little bit about the blood supply and nerve supply, the vessels and nerves enter the large salivary glands at a hilum and gradually branch into lobules. A rich vascular and nerve plexus surrounds the secretory and duct components of each lobule. The capillaries surrounding the secretory units provide fluid important for saliva production, which is stimulated by the autonomic nervous system. Parasympathetic stimulation, usually elicited through the smell or taste of foods, provokes a copious watery secretion with relatively little organic content. Sympathetic stimulation inhibits watery secretion and produces the potential for dry mouth, often associated with anxiety. Okay, so till now we have discussed about the general structure of the salivary glands. To recap, we have discussed the components and its details, the arrangement of the components in general, and the blood and nerve supply to the gland. In the next session, I will be discussing on the specific features for each of the salivary glands. See you there. Join my Facebook community, link in the description. Like, share and subscribe. Thank you for watching.